DNA is the genetic material. Transformation. 1. The conversion of a normal animal cell to a cancerous cell. 2. A change in genotype and phenotype due to the assimilation of external DNA by a cell. When the external DNA is from a member of a different species, transformation results in horizontal gene transfer. Bacteriophages. A virus that infects bacteria, also called a phage. Virus. An infectious particle incapable of replicating outside of a cell, consisting of an RNA or DNA genome surrounded by a protein coat capsid, and, for some viruses, a membranous envelope. Double helix. The form of native DNA, referring to its two adjacent antiparallel polynucleotide strands wound around an imaginary axis into a spiral shape. Antiparallel. Referring to the arrangement of the sugar phosphate backbones in a DNA double helix, they run in opposite 5-3 directions. Many proteins work together in DNA replication and repair. Semiconservative model. Type of DNA replication in which the replicated double helix consists of one old strand, derived from the parental molecule, and one newly made strand. Origins of replication. Site where the replication of a DNA molecule begins, consisting of a specific sequence of nucleotides. Replication fork. A Y-shaped region on a replicating DNA molecule where the parental strands are being unwound and new strands are being synthesized. Helicases. An enzyme that untwists the double helix of DNA at replication forks, separating the two strands and making them available as template strands. Single-strand binding proteins. A protein that binds to the unpaired DNA strands during DNA replication, stabilizing them and holding them apart while they serve as templates for the synthesis of complementary strands of DNA. Topoisomerase. A protein that breaks, swivels, and rejoins DNA strands. During DNA replication, topoisomerase helps to relieve strain in the double helix ahead of the replication fork. Primer. A short stretch of RNA with a free 3' bound by complementary base pairing to the template strand and elongated with DNA nucleotides during DNA replication. Primes. An enzyme that joins RNA nucleotides to make a primer during DNA replication, using the parented DNA strand as a template. DNA polymerase, an enzyme that catalyzes the elongation of new DNA, for example, at a replication fork, by the addition of nucleotides to the 3' end of an existing chain. There are several different DNA polymerases. DNA polymerase III and DNA polymerase I play major roles in DNA replication in E. coli. Leading strand. The new complementary DNA strand synthesized continuously along the template strand toward the replication fork in the mandatory 5'3" direction. Lagging strand. A discontinuously synthesized DNA strand that elongates by means of Okazaki fragments, each synthesized in a 5'3" direction away from the replication fork. Okazaki fragments. A short segment of DNA synthesized away from the replication fork on a template strand during DNA replication. Many such segments are joined together to make up the lagging strand of newly synthesized DNA. MisMatch repair. The cellular process that uses specific enzymes to remove and replace incorrectly paired nucleotides. Nuclease. An enzyme that cuts DNA or RNA, either removing one or a few bases or hydrolyzing the DNA or RNA completely into its component nucleotides. Nucleotide excision repair. A repair system that removes and then correctly replaces a damaged segment of DNA using the undamaged strand as a guide. Telomeres. The tandemly repetitive DNA at the end of a eukaryotic chromosome's DNA molecule. Telomeres protect the organism's genes from being eroded during successive rounds of replication. Telomerase. An enzyme that catalyzes the lengthening of telomeres in eukaryotic germ cells. A chromosome consists of a DNA molecule packed together with proteins. Heterochromatin. Eukaryotic chromatin that remains highly compacted during interphase and is generally not transcribed. Euchromatin. The less condensed form of eukaryotic chromatin that is available for transcription. Word roots. Helic. A spiral helicase, an enzyme that untwists the double helix of DNA at the replication forks. Word roots. Liga. Bound or tied DNA ligase, a linking enzyme for DNA replication. Word roots. Phage. To eat bacteriophages, viruses that infect bacteria. Word roots. Semi. Semiconservative model, type of DNA replication in which the replicated double helix consists of one old strand, derived from the parent molecule, and one newly made strand. Word roots: telos, an end, telomere, the protective structure at each end of a eukaryotic chromosome. Word roots: trans, a cross-transformation, a phenomenon in which external DNA is assimilated by a cell.